Okay. All right. So hopefully you can now see the PowerPoint.
Yeah.
Fantastic. All right. So I may not get the whole way through this. That's probably okay. I'll do a screencast around it if I need to, because it's going to help you a bit with what I'm asking you to do for the holiday homework. I'll obviously explain that as well as part of what we do before I let you go today, and then, show you my dog. The learning intention is to learn about the second line of immunity. So this is chapter 7C. And the goal is to understand the role of cells of innate immunity. So there's a number of different cell types. They're named in the study design, so you're going to need to know them and specifically what they do. Non-specific humoral defenses. Humoral just means chemical, all right? So, or non-cell, basically, and we'll touch on those. And then there's 3 stages of information that we need to walk you through. And that bit in yellow, that's what your study design says. It's pretty much just rewording what I said, except it's naming those cells specifically. So the innate immune response, including the steps in inflammation, the characteristics and roles of macrophages, neutrophils, dendritic cells, eosinophils, killer cells, and mast cells. So it names all those cells. You're going to need to know what they do. I'll walk you through that, and we'll set up some summaries around that too. You also need to know about complement proteins and interferons. The level of detail in this study design and in this textbook are less than what I've encountered in the past for those last couple. That's probably positive. Complement is exceptionally complicated. You do not need to know the complications. You just need to know the basics of it, thank God. I had to learn it at uni and I had to memorize the whole cascade. I do have a slide of the whole cascade. You do not need to know the whole cascade though, but I'll show you when we get to it. So this is what we're talking about here, this second line. It is still innate, so you are basically born with it. It is not adaptive or acquired. It's what you're born with, and it is still non-specific, which is really important. So it doesn't matter whether it's a bacteria, a virus, a prion, an allergen. It doesn't matter. These are just general defenses that will kick into action as soon as an invader is detected, whatever it is. Over here, it takes a couple of weeks and it's very specific, like very, very specific. Sort of, how enzymes and substrates are very specific. It's that level of specificity, but that's chapter 7D. We'll talk about that later. This is from the textbook that I've used previously. It just sort of spells out much the same thing, it just with a little bit more detail, breaking it down into, like over here, humoral or antibody mediated. But this is what we've already touched on, is the mechanical, chemical or physical, chemical and microbiological barriers. What we're really looking at here is this cellular response in terms of phagocytosis and the triggering of inflammation. So that's sort of what we're looking at here. I've touched on this already. 2 broad categories, cellular and non-cellular or humoral. And starting with cellular, this is from your textbook. The ones at the top here that are highlighted in red, those are the cells that are named in your study design. Okay, down the bottom here, it's got a key that is helping to spell out what each of them do. Eosinophils and mast cells, we'll touch briefly on what they do. Mast cells we look at a lot more when we do allergies and the way allergies work. One of the key things you need to know is about phagocytosis, which I don't think we've studied before, but we've probably talked about it. Do you remember we've talked about phagocytosis as an example of something? Do you remember what it's an example of? It's bulk transport across a membrane. Does that help at all in terms of what the process would be? No? Okay, it will. I think once you see it, you'll go, oh yeah, I get that. No, I hope. I suspect that that'll be the case. So these 3 with the blue triangle are phagocytes. The 2 with stars are very important cells. They used to get a specific mention in the study design. They don't anymore. So I don't know if the emphasis is as great anymore, but they're called antigen presenting cells. Really, really important in terms of your adaptive immune response. So they sort of blend together a little bit, but we'll talk about that when we get into it. But they have a very important role to play. This is going to help you with some work that I'm going to ask you to do over the holidays. So this basically shows you innate immune response. Here's those cells that we talked about. You'll see there's a little bit of crossover here in the middle. Natural killer cells is one of the cells that we need to talk about as part of the innate immune response. And then you've got your adaptive, which is predominantly your B cells that produce antibodies. and there's T cells, which are cytotoxic T cells, and we'll talk about those more. This video will explain phagocytosis, I'm hoping. I don't know if there'll be sound or if it's going to let me trigger sound. I'm going to find out. Nope. Can you hear that?
No.
Excellent. So how do I fix that? What does that button do? Nope. I don't want subtitles. Make that go away. Okay, that was good. All I want is sound. What's that do? Oh no, that was bad. How do I undo that? Okay. I can hear what she's saying. It doesn't really matter too much. I'll explain it and I'll have to try and talk. I'll mute that video so I can talk over the top of it. So what it's pointing out there is a neutrophil. It's a white blood cell, so it's one of the phagocytes, okay? Red blood cells there, just for comparison. There's some bacteria there. What you'll see in this video is that the bacteria will... Okay, so what you'll see is the bacteria will start moving. It looks like it's pursuing the bacteria, which it's using cytokines to actually be attracted towards it. They literally do move through the bloodstream like that. It will wrap it in pseudopods like that. So it's now surrounded it, it's engulfed it. It will bind the bacteria with a lysozyme, which is full of digestive enzymes, and it will break down the bacteria and destroy it. All right. The only thing you missed is that someone who has warped sense of humor played some music like it's from Jaws, and at the end the white cell burps. That's pretty much what you missed. You didn't miss a hell of a lot. All right. So it engulfs the bacteria. That's what phagocytosis is. It will engulf the bacteria. It immerses it in digestive enzymes. So it closes it in a vacuole. That vacuole fuses with a lysosome. It inserts digestive enzymes and it kills the bacteria and breaks it down. That's the process of phagocytosis, and we'll go through that in a second. Your types of phagocytes, they're the ones with the blue triangles in this diagram, and the neutrophils, which was in that video, macrophages and dendritic cells. And these are the stages of phagocytosis. So you've got the pathogen, it makes this pseudopod, it encloses it in a vacuole, your lysosome fuses with it with the digestive enzymes, breaks it down, and then spits it out. Thinking about membrane transport, what is that process? Starting with E.
Do not tell me you go for Carlton.
You
Did not know that.
You know that I go for Carlton, but
I didn't actually.
We've talked about it before, and you slagged me off for it before. So, you know, I remember because I'm still hurt by it. That's okay.
You need to throw that mug out.
Or possibly my shirt. So, anyway, so we're moving on. I thought it was actually the shirt that gave it away, but yeah, I forgot about the coffee cup. What's the process? Starting with E. When stuff is engulfed inside a cell, what's the name of that process?
Endo
what?
Cytosis.
Yeah, endocytosis. Good. All right, this is touching on the antigen presenting cells. So your dendritic cells and your macrophages are phagocytes, right? So they will do exactly what you just saw, but they have this extra step. So you remember MHC1 and MHC2? Yeah. MHC, where do I find the MHC2 molecules?
Immune cells.
On immune cells. So things like macrophages and dendritic cells. So this one's from your textbook. Same process. Here's your pathogen, engulfed, digested by enzymes. But instead of it just throwing all the stuff out, so in this diagram, it's throwing everything out. In this diagram, what it's showing you is that it breaks down fragments of the pathogen and actually binds them on the surface with MHC2. So it actually basically holds them up like a flag saying, here it is, this is the pathogen, I want you to come and do something about it. That's literally what antigen presentation is, taking parts of the pathogen. You've destroyed the pathogen, but you want to create an adaptive immune response. So you hold up part of the pathogen to the immune system and say, look, I found this, come and kill it, come and deal with it. That's what antigen presentation is. We'll go into how that works with the adaptive immune system when we look at the adaptive immune system. All you need to really understand at the moment, 3 types of phagocytes: neutrophils, macrophages, dendritic cells. 2 of them, the macrophages and dendritic cells, are capable of displaying parts of the antigen together with their MHC2 markers. Is that okay? That's not okay? Which bit's confusing you?
All of it.
It often seems to start that way. So do I need to explain it better, or do you just need time to review it and then come back
Just
to
keep going.
Okay, that's not an answer to the question though. Do you need time to just review it, come back to it, and let it embed for you?
Yeah.
Okay. Because we can go over this stuff like we've done with a lot of other stuff. It'll be easier when I'm at school. But we can do that, okay?
Yeah.
So don't stress. All right, hopefully this is not too confusing, although I'm worried that it might be. We'll see how we go. All right, so natural killer cells, and one of the other cell types you need to know about, their job is to actually kill self cells. So infected or damaged or stressed is the way they word it, stressed self cells. The 2 forms of stress typically are they're infected with something. What sort of pathogen is likely to infect the inside of one of our cells?
Viruses.
Yeah, viruses, intracellular parasites. So viruses are likely to cause infection that trigger this. The other typical cause of stress is cancer. They're the 2 that they give as an example. There's 2 receptors, the MHC1 markers. Remember, all of your nucleated cells have MHC1, and then there's killer activation receptors, and that'll make sense when I show you the diagram, I hope. I've already answered that, so I shouldn't have done that. MHC1 are on all nucleated red cells, on all nucleated cells, and MHC2 markers are only on immune cells. So this is the way they work. So if you've got a normal cell, remember I described MHC1 markers as like that handshake. You've got these natural killer cells, they're floating around and they're checking all the time to make sure that everything's under control and everything's normal. So what they're looking for is this MHC1 marker that's on the nucleated cell, right? There's an activating ligand. The activating ligand will actually trigger the natural killer cell to kill it. But if it hits the MHC marker, if it picks up the MHC marker, it recognizes it as self, it recognizes it as healthy, and it will leave it alone. So it will bind like it has in this image and then just let go. That makes sense?
Mm-hmm.
Yep. Over here, an infected cell or cancer cell, whatever, the MHC marker is damaged or completely gone, right? So now there is no MHC marker. The activating ligand is still present. So the natural killer cell will bind to this activator, but it's looking for that handshake and it's not getting it. So now it's going to recognize the cell as being damaged or diseased and it will kill it. Does that make sense?
How does it kill it?
I'm going to show you that in a second. It sort of hints at it here. These granules that are inside it get released inside the cell, and it basically enzymes that poison the cell and kill it. It basically injects poison directly into the cell. Is the easiest way to describe it. But the biggest thing is the MHC marker. So if your MHC1 markers are intact and recognized by your immune system, the natural killer cells leave it alone. If the MHC marker is missing, then it's going to recognize it as needing to die and it will kill it. That's really what you need to know. Is that okay?
Mm-hmm.
Yep. This one's more complicated. It's from your textbook. It's telling you exactly the same thing. This one is not from your textbook. I think it's much simpler. Like it's normal cell, happy MHC, everything's normal. MHC missing, abnormal cell, kill it, make it die. Like that's simple. This one is telling you the same thing, but there's, I don't know, it's just a busier diagram, and I'm going to skip that because I think it's too complicated. And then what's happening here is degranulation. The 2 cells here are bound together and the natural killer cell has been activated. Don't stress too much about this diagram. This is just answering that question that you just asked, Flick, which is the natural killer cell will start by inserting these proteins to make a pore in the surface of the target cell. And then it releases granzyme, which is an enzyme that is effectively toxic to the cell. So it literally will pass that through into the pore, enters the inside of the cell, kills the cell. Okay? Yep, great. The last 2 cell types that you need to know about, mast cells, which we'll talk about more later. But mast cells will be activated when they detect injury or in response to allergy. They release histamines. So Paige, yesterday you said you get hay fever. I imagine you take antihistamines to keep that under control.
Yeah.
This is why. These guys here are the ones that have the granules in them are called histamines. They trigger things like the runny nose, the watery eyes, the sore throat, the closing of the throat, all that kind of stuff that you experience with hay fever. They do other stuff as well, but that's what they do. When you take antihistamines, you are counteracting the effect of those with hay fever. But we'll talk about those more later, but they have a key role in inflammation, and that comes up in this chapter. So just lock that bit of information away, that they release histamines. We'll talk about that in a sec. Eosinophils are basically another granular cell. Their granules release particular enzymes like DNAs, RNAs. Anything that ends in A's typically means it breaks it down. So it's DNAs, it breaks down DNA. RNAs, it breaks down RNA. And protease, it breaks down protein. So they have granules that actually break down the pathogen. They're typically used against big pathogens, and having worked in hematology where we look at people's blood, if you have a worm infection or a parasite infection, so they're bigger than, like they're worms and stuff like that, right? They can't be phagocytosed, they're too big, because, you know, a phagocyte's a single cell, a worm is a whole bunch of cells stuck together. So they can't phagocytose it. That's typically when eosinophils kick into action. They attack the bigger pathogens and release these toxins that kill it. Is that okay?
Mm-hmm.
Yeah. I don't expect you to remember all of it. It's okay. How long have we got? I want to make sure that I don't just rush through stuff and bombard you and then it won't make any sense. May not make sense anyway, but we're going to look at ways to consolidate that, I promise. Okay. So that's the cell types. All right. So again, if you go through your study design, it says those are the cells you need to know. Neutrophil, macrophage, dendritic cell are phagocytic cells. Then you've got your mast cells, your eosinophils, and your natural killer cell. All right. So those are the cell types you need to know. The non-cellular responses, there's two key protein or groups of proteins that are involved. And again, they're named. You needed to know interferons. All you need to know about interferons really is that a virally infected cell, so remember viruses enter a cell and then reproduce. If a virus enters a cell, it triggers the release of interferons. Interferons then circulate to nearby cells. They send a message. It's basically like a distress signal saying, hey, I'm infected, just warning you, you might want to protect yourself. It literally causes the cell to make changes to its cell wall so that it's harder for a virus to infect the next healthy cell. So it doesn't do anything to protect the cell that's already infected. It's already been infected. It actually just sends out a warning to surrounding cells to say, hey, there's something going on here. You might want to, you know, draw up the drawbridge and protect yourself. Is that okay?
Yep.
So that's what interferons do. If you want to remember it, they interfere with the virus's ability to get inside the cell. That's kind of what they do. Complement. There's three key things that complement does, which is opsonization, which is like planting flags on the surface of the pathogen to say, hey, come and phagocytose this, come and kill it. Inflammation, which we're going to talk about, and the membrane attack complex, which is cell lysis. First, we need to talk about complement. So there's a complement for you. And there's another one. I quite like that one.
I like ladybugs.
Yeah, but, you know, tigers are better. You've got to be honest.
No.
Okay, well, you're a ladybug then. And Flick, you can be a tiger if you prefer tigers. All right.
Yes.
Pick which animal you're happy with and we'll move on. Because that's not what complement is. That's complement. You do not need to know that at all. Okay, that was when I said it's really complicated and I had to memorize that for uni, that I had to be able to reproduce that. You do not. I do not remember it now. I presumably remembered it for the exam because I passed, but it's ridiculous. You just look it up, right? Even this level, which is a much more simplified diagram, you don't need to know this level of detail. What you do want to know is that complement can recruit immune cells, and that's triggering inflammation. That it can opsonize a pathogen, which triggers or enhances, as it says there, phagocytosis. Or it can make the membrane attack complex, which causes lysis of the cell. So we'll quickly walk you through those. So this is all from your textbook, all on page 379. Opsonization is just a fancy word that literally means sticking a flag on the pathogen. All right. So complement will be triggered in response to any foreign antigen. Again, remember, it's non-specific. It's not this E. coli or this particular fungus. It's not doing that. Anything that's not supposed to be there, it will trigger complement. Complement will stick to the surface of the pathogen, and it acts like a big red flag on the surface of the pathogen saying, hey, come and kill this. And that's really what it is. So you've got C3b, which is one of the complement proteins. You won't need to remember that. Just know that complement binds to the surface of the pathogen. And macrophages have complement receptors that actually stick to the complement. So it literally will attract the macrophage. It sticks to the complement, which is already stuck to the pathogen, and it's more likely to lead to phagocytosis. Is that okay?
Mm-hmm.
Yep. All right. Chemotaxis, which is literally just movement in response to chemicals. All right. So remember, one of the outcomes of complement is recruiting immune cells. So complement literally sends a signal out to the immune cells and say, hey, come here, come and clean this up. Because if you imagine, and you're going to see this in a second, hopefully, as long as I can get through it in time, if you injure your arm, for example, or I think the example in your textbook is you're cutting some wood, you get a splinter. All right. Splinter penetrates the skin. Your body's quite big, your splinter's quite small. How do the immune cells know where to go to actually start doing stuff? It's as a result of these chemokines or cytokines. The cytokines get released, which are just a signaling molecule, and say, hey, come over here. Complement plays a part in that. It's a chemical signal that says, this is where I need you to be, and then the immune cells flood that area. Is that okay? Yep. And that's all that says, so don't worry about that. And then membrane attack complex. Some of the complement proteins will stick together and stick a hole in the cell wall of the pathogen, literally lysing it. Okay, so it sticks a hole in the cell wall and it bursts open. That's what the membrane attack complex is. Again, you don't need to know the specific complement proteins. These C3, C5b, C6, all of that, they're the subcategories of the proteins. Don't worry about that. But these various complement proteins will basically form a pore, literally punching a hole in the membrane of the pathogen, bursting it open. So it causes lysis, kills the pathogen. We good with that? It's a lot to remember, I know, right? Non-specific response, again, is fever. We've touched on that already. The goal of fever is to basically cook the pathogen. A lot of pathogens won't survive if your body temperature goes above 37 degrees. So your body deliberately makes you hot so that you are hopefully killing the pathogen. Obviously, that's quite dangerous if that's for a prolonged period, because if you get too hot, you know that your enzymes denature, all your biochemical processes stop, and then you die, which also kills the pathogen, but it's not really very effective at keeping you alive. So, but fever is a non-specific response again. It doesn't matter what the pathogen is, that's something that your body's going to try to protect you. Yeah, I can get through that. Okay, inflammation. We'll do this really quickly. That's from an article saying, oh, inflammation's all bad and it's going to destroy you and pull you apart. Extensive inflammation and chronic inflammation is bad. It will cause damage over time, but inflammation is a normal response to invasion with a pathogen. It's a really important response, and it's in that second line of defenses. So once a pathogen, because remember, your first line is trying to keep everything out. That's the ultimate goal, keep everything out. The second line is saying, all right, that first line's failed. The pathogen's now inside me. Now what do we do? And that's where inflammation kicks in. A lot of what you've seen so far, learning about the cells and what they do, learning about phagocytosis and how it cleans stuff up, learning about mast cells and the histamines, it actually all ties in with this last little bit of this chapter, which is about inflammation. So inflammation is damaging potentially, but it is a normal process. It occurs quickly within minutes or hours of an initial injury. And the goal is to localize and prevent pathogen spread. So again, if you've got a splinter and it's stuck in your skin and you've inserted bacteria inside yourself, which will happen potentially as part of that, you've got bacteria in you now. It doesn't want them to spread through your whole body. It wants to keep them restricted in that immediate area so they can be killed and mopped up. So eliminate the pathogen, remove the debris, and repair the tissue and restore a normal state. And it does that across 3 stages. Again, I need to work out how to play the video so you can hear it, but I don't have a button that lets me do that, which is bloody annoying. So I'm going to skip that for now. And then I'm going to send you the video and you guys can watch that as part of your revision. Would be a really good idea. And I'm going to play on Teams to work out how to play a video with sound. But anyway, we'll work that out. All right, so the 3 stages of inflammation. Initiation. So here's that splinter. It's punctured your skin. It's inserted some bacteria as part of it. As you break the skin, it releases these cytokines. Again, cytokines are signaling molecules that are sending a signal saying, hey, there's something going on here. This is where I need you to be. This is where I need you to act. Here are your mast cells. Your mast cells have been recruited to the area and they degranulate. They release histamine. Histamine then causes what they call leaky blood vessels. So down here is your blood vessel. You've got your white blood cells, your phagocytes, all right, so neutrophils, macrophages, that kind of thing, and your red blood cells, obviously. If you think about inflammation and stop and think about what's going on, like if you took your arm and scratch, don't actually do it, but if you scratch quite hard along your forearm, what do you notice happens?
It raises.
Yeah, it gets raised. All right, we're going to come to that. What else does it do?
Goes red.
Goes red, yep. And depending on how bad it is, you might also feel heat. All of that is related to this inflammation response. That is literally an inflammatory response. So the initial injury has triggered off your histamines. Your histamines then lead to this next stage, which is vasodilation. So the blood vessels dilate and they allow the white blood cells, these white blood cells, through gaps in the blood vessel, right? And the phagocytes will start phagocytosing any bacteria that are there. Fluid will also penetrate, leads to swelling. Right, so the swelling is actually literally this leaky blood vessel stage, that there's fluid leaking into the injury area to allow the cells in there and everything else that's required, the complement and all the other stuff to start cleaning stuff up. Does that make sense? So when you see swelling, it's actually because there's fluid penetrating the site. Also because you've got increased blood flow to the area, that's what leads to it going red. And because you've got blood going to the area, blood is warm, it also increases heat. So if you stop and think about, if you think of inflammation as I've scratched really hard along my forearm, and you think, okay, well, it swells, it goes red, it gets hot. Why? That's going to help you remember that while the blood vessels are leaking, it fills with fluid, there's blood flowing to the area, it's leading to heat, redness, and swelling. Does that make sense?
Mm-hmm.
Just trying to think of ways to make it easier for you to remember. So your first stage there is initiation, then vasodilation, which allows blood, fluid, and the white blood cells into the area. Migration, so you've got complement, you've got your phagocytes all infiltrating the area, they're migrating to the site, and they start cleaning everything up. So the stuff we already talked about, phagocytosing the bacteria, complement will opsonize the bacteria to increase the likelihood the phagocytes will kill it. You'll have complement lysing the bacteria, etc., etc. And that's a summary basically of what happens there. Tissue damage and infection releases the cytokines. Histamine is triggered. The histamine leads to vasodilation with increased blood flow, leading to redness, swelling, heat, and pain. Increased blood vessel permeability, which allows in the phagocytes and complement. They clean up the bacteria, they clean up any damage, and then ultimately you get resolution, which is tissue repair and infection clearance. Is that okay? That's a lot to take in, and I know it is. All right, so that's actually a summary from page 382 of your textbook about inflammation. I've sort of done it out of order because it made sense to put it in here. Just before that, there's also this summary of those are the cells named in your study design. This is what you need to know about them.
Is that all we need to know about them?
Pretty much, yep. Obviously, underneath that, you need to know what phagocytosis is, or you need to know how a natural killer cell will identify which cells need to be killed and which ones don't. But in basic terms, yeah, that's what you need to know. And then same for your non-cellular. What does an interferon do? What does the complement proteins do? And what is fever aiming to do? So those two, that one on page 382 and that one on page 382 are good summaries of what you need to know. And that's pretty much it for that. I didn't know that we'd get through the whole chapter, but it looks like we did. Oh, look at that. You've disappeared. Let me bring you back. I know that's a lot of information, so I don't expect you to sit there and go, yes, that all makes perfect sense and it was really easy. But does it make some sense? And with revision, do you think that you can consolidate that and understand it?
Yep.
Do you play poker, Paige?
What?
Do you play poker?
No.
That's probably for the best because you don't have the world's most tremendous poker face. Because I asked you that question and the look on your face was like, no, I do not believe anything that you just said. And that's okay, as long as we work through it. I'm going to post this in a second. This is your holiday homework. This is something similar to what we've done together countless times before. But what I'm hoping we can do is I haven't done all of this. I've thrown together a very brief example to show you what I'm aiming for. And it's by no means complete. What I'm hoping you can do is consolidate the stuff that we've done so far. So that is this first line of defense here and the second line of defense here. All right. Those are the two that we've actually covered together in class. What I'd like you to also do is look at chapter 7D and look at the third line of defense and create a summary around that.
So
to give you an idea
as well.
Sorry?
Do you have to do 7E as well?
Yes, because it talks about the lymphatic system. So lymphatic system is going to be important in terms of understanding this third line of defense. So reading through those and making a bit of a summary. But the goal is to pull out the key information, right? So first line of defense is all about your barriers, right? So whether you use this image or not, I don't care. It doesn't matter. Whether you do it electronically or not, I don't care. You can just sit there with pen and paper and crayons and make it with Play-Doh. Like, I really don't care how it's done. I would just ideally want you to have a summary that you put together, which means that you've pulled out the key information and identified what you need to know, and made a summary that you can refer back to as you do revision. All right. So first line of defense is barriers. And so what I've done is, all right, we've got physical barriers. What does it do? Prevents entry, skin and mucous membranes. Barriers here, chemical, damages or destroys pathogens, and some examples. Microbiological, competes for nutrients in space, and some examples. Under the second line of defense, which is what we've just covered, cellular and then non-cellular. Cellular, I've listed the cell types and what their function is and what they do. Then I would go further and go, okay, well, phagocytosis, what is that? Natural killer cells, what do they actually do? Right now, I've just written explain steps. Obviously, where I've said that, I would prefer that you explain the steps. And then over here is just a summary of what I'm aiming for. This is a starting example. Your job that probably should say is to create wow, I've really proofread that well. I'm going to blame COVID. It's fine. Your job is to write English better than me.
I cannot fix it.
So your job is to make a detailed outline of the 3 stages using revision of what we've done and also reading ahead to start getting it. We'll go through those together when we come back, chapter 7, D and E. Right, but the goal will be that you'll come in next term having an understanding of that already, maybe not a full understanding, but some understanding, and then we'll go through it together and be able to fill in any gaps that you've got. So you're not starting from scratch next term. Is that fair? Yep. This little bit here, and I have very little doubt that either of you will do this, I sincerely hope. Don't come in next term and say, I didn't do it because I didn't get it. If you didn't get it, message me, okay? If you really stuck and you want to catch up over the holidays, tell me that that's what you want to do. All right. I'll be at school a couple of days anyway, because apparently my new job's like really hard and stuff, and I need to learn some stuff. So I will be at school a couple of times through the holidays.
Close that door.
And I'm more than happy. I'm going to say that just messed with my head because I'm looking at you looking sort of past me there, Flick, at Jet, who was obviously then behind Paige, and it's like catching multiple angles at the same time. So yeah, so the goal will be that you do this, but please, like if you want to catch up on the holidays and do stuff, I'm sure you all have better things to do. Go down and catch up with penguins again, Paige, I don't know. But I'm sure you've got better things to do than catch up with me. But if you want to, let's do that, okay? Because we're getting to the pointy end now. Let's make sure that you come in next term ready with this stuff consolidated. Certainly I can meet with you guys again next term and sit down with you and go through stuff like we've done with photosynthesis and respiration, and I'm happy to do that. But yeah, I want to make sure that you don't sort of come in going, oh my God, I'm completely lost still, and none of this stuff we've already covered makes sense, and I haven't looked at the other stuff. That's really important. Is that okay?
Yeah.